Cell division, genetics, and molecular biology. Chromosomes and the cell cycle. The characteristics of each individual are coded for by the section of DNA called genes. Each gene codes for one trait, such as hair color. Chromosomes are long strings of genes lined up in a specific sequence. The more genes in a species, the greater the number of chromosomes need. Humans have approximately 20,000 genes which fit onto 23 chromosomes. However, because humans are sexually reproducing organisms, the normal human chromosome number is 46. The guarantee of sexual reproduction is that each parent will contribute information towards each trait. Thus, both mother and father provide the zygote with a full set of 23 chromosomes in their egg and sperm. The zygote and the offspring that forms from it have two complete sets of genetic information. Whether the offspring will actually express the mother's gene, example dark, or father's, example light, depend on the rule of dominance and recessiveness. Two chromosomes in a nucleus that have the same gene sequence and come from each parent are called a homologous pair. All cells that have homologous pairs are said to be diploid or have the 2N chromosome number, which in human is 46. All cells that have derived from the zygote by mitosis are also 2N. If all zygotes have the diploid number of chromosomes, then egg and sperm cannot. They must have only 23. Diploid cells in the ovary or testis undergo meiosis to produce cells with the haploid or N chromosome number, which in humans is 23. When a 2N ovary or testis cell undergoes meiosis to have the chromosome number, only one chromosome of each homologous pair is allowed to enter a gamete. Whether that chromosome is maternal or paternal is completely random. Each haploid sperm will therefore be a unique combination of the man's maternal and paternal chromosomes. Each haploid egg will be a unique combination of the woman parent's chromosomes. When a sperm and egg unite in fertilization, the diploid chromosome number is restored in the zygote and every cell that is derived from it in mitosis. The 20K genes expressed in an offspring are a unique combination that is never repeated in any individual except identical twins. Mitosis is self-division that produces two cells that are genetically identical to each other and to the mother cell. Mitosis occurs when an organism grows or needs to repair itself. The rate of mitosis is mostly dependent on an individual's age. When one is very young, the rate of mitosis is far greater than the rate of cell death. Extremely rapid rates of mitosis are typical of cancers. The cell cycle interphase mitosis and cytokinesis. The period from one cell division to the next is called a cell cycle, which is divided into three stages: interphase, mitosis, and cytokinesis. One cell cycle can be hours or years long, depending on the type of cell. Cells spend most of each cycle in interphase, during which the cell carries out its normal functions and grows larger. Interphase can be divided into three phases: G1, S, and G2. During gap 1, G1 phase, the cell grows. Chromosomes are not visible during interphase. At this point, oink, they are long strings of uncondensed DNA called chromatin. During the S or synthesis phase, the cell begins to prepare for cell division by replicating the DNA. The 46 strings of chromatin, 23 from each parent, are replicated. Each chromatin string and its replicate become buttoned together by a centromere and are referred to as sister chromatids, det. In gap 2, G2 phase, the cell continues growing until its volume to surface area ratio becomes too large and a cell division becomes necessary, declutter or purge. The soul of mitosis is to separate the 46 sets of sister chromatids into two complete sets, One chromatid from each pair of sister chromatids should go to each daughter cell. Mitosis has 4 stages: prophase, metaphase, anaphase, and telophase. Prophase begins when the nuclear membrane and nucleolus disappear, making room for an organizing spindle which appears shortly. The DNA, which to this point was invisible chromatin, suddenly becomes visible as it supercoils, condenses, to form solid chromosomes, which are much easier to move around the cell. 2 sister chromatids joined by a centromere, making 1 chromosome. During prophase, 2 centrioles can be seen migrating toward the cell poles. As the centrioles move apart, spindle fibers form, stretching between the centrioles to form a spindle. In metaphase, the chromosomes migrate to the metaphase plate and line up along the cell equator. A spindle fiber attaches to each centromere. In anaphase, the centromeres divide so each sister chromatid has its own centromere. Because the definition of a chromosome is any DNA held together by a centromere, each sister chromatid is now its own chromosome. As anaphase continues, the spindle fibers attach to the centromeres and proceed to pull the sister chromatids apart, forming a cluster of chromosomes at each pole. The last phase of mitosis is telophase, during which a nuclear membrane reforms around each of the 2 clusters of chromosomes. Chromosomes decondense to form chromatin and become invisible again. Mitosis is then complete. At this point, the mother cell is still intact but has 2 nuclei. The cell cytoplasm is split into 2 separate cells in the process of cytokinesis. In animal cells, cytokinesis occurs when the cell pinches in the middle to form a cell furrow. In plant cells, the cytoplasm splits when a cell or division plate forms down the middle of the cell. Once cytokinesis is complete, the 2 identical daughter cells enter interphase, beginning a new cell cycle.